All right, well, welcome everybody. Thanks so much for joining us to our second edition of our STEAM program, STEAM 2.0, as we affectionately call it here on campus. And today we have a great hour of conversation with two different artists, artists in different ways that think about science in different ways. And I think this is going to be a great pairing. So just to begin with, though, I want to just thank a few folks for making this possible. Primarily among them, I want to thank Ana Garcia, one of our faculty members in chemistry and earth sciences, for really being the leader of this whole endeavor. She kind of got it all started and continues to just volunteer crazy amounts of time to make sure all these things come together. So I want to thank Ana. I also want to thank Elizabeth Thomas from the library, who put together all the websites, put together the art exhibits, and all things connected to the STEAM 2.0 program. And now I'd like to also move on to today's speakers. And we have two of them, as I mentioned. We have Abrian Curington, illustrator and cartographer. We're going to be seeing some of her amazing work. Let me just move a screen here so I can see my own. So a graduate from Western Washington University. We have a lot of graduates from Western Washington around the parts of Monterey, and has been incorporating her childhood love of science into work in creating maps and weaving information among her different works. And that actually is something that I think is going to be a nice unifying thread between what Steve talks about and what Abrian talks about, is how do you display information? How do you explain science? How do you tell stories? And we're going to see that talked about in a couple different ways here today. So we're going to start off with Abrian, and then once maybe about 20-25 minutes go by, we'll move into Steve's portion of the talk. And Steve is a researcher at Monterey Aquarium Research Institute, which is here in Moss Landing in California. He is also affiliated with UC Santa Cruz, and he has a long, illustrious career specializing in lots of gelatinous, squishy-like creatures like jellyfish and ctenophores that are in the water column and the deep sea, and uses a lot of cool toys and tools to explore these depths and bring these animals up for photography and other studies. He is a worldwide leader in bioluminescence and operates a very popular web page for bioluminescence, and has written a number of other compelling pieces, which also includes a popular textbook on practical computing. So we'll perhaps, he'll weave some stories perhaps between many of these subjects in his piece today. So I want to thank you again all for coming. And we're going to begin now. I'm just double checking that our student Stephanie has not brought it. Okay, so we're going to move from there, and I'm going to invite Abrian to begin sharing her screen, and we'll kick it off. So thanks a lot for joining us, everybody.
All right, can you guys hear me?
We can hear you, yeah.
Okay, for some reason, Zoom kept kind of half freezing itself and half not. So
okay.
I will not try to go into video again. I will just share my screen.
Okay.
Oh no, looks like I'm not a co-participant anymore.
You were, oh, maybe let me do that again for you. So now try again right now, that should work.
Perfect. Okay, so this is just some things to look at as I speak. I am Abrian Curington. I'm a visual storyteller and cartographer, which means that I tell stories using illustrations, videos, or comics, and that I make maps using both GIS data and fictional data. So I do a little bit of everything, and me and Tolkien would be best buddies. However, it is a common misconception that I'm actually a scientist. I'm a nerd for sure. I grew up loving deep sea and deep space, stargazing, collecting shells and rocks. I'd come home with pockets full of rocks and acorns. But I never actually wanted to be a scientist, my profession, because I wanted to be an artist. Besides, if I were to become a scientist, I would have to specialize, and I wanted to learn about everything. So how did I, a freelance artist, end up searching for meteorites in the deep sea, which was a perfect blend of my most two favorite topics? And why is it important to have artists and science communicators in the field as discoveries are being made? Now, science and art have actually been intertwined for most of our history. Scientists would not only keep notes about their work, but they would also draw their findings as well. I don't know if you remember back to elementary school, but I remember having to draw my observations of like a decomposing banana. I'd have to go check on it every once in a while and like, okay, well, there's some brown up top and it's oozing down below. So I had to both write and draw my findings. But at some point, the idea of art and science started to become compartmentalized. Art is over here being expressive and lawless, which there are rules. People like to say there aren't, but there are. And science is over on the other side being cold and data-driven. But recently, there has been greater awareness around the power of science communication and its use to make the public more aware of scientific discoveries. So now, not only are the sciences starting to use clearer language to communicate their findings, but they're also utilizing visual storytelling to spark curiosity, so the public can become more interested in the cause that they're studying. And visual storytellers, like myself, play on the idea that the more senses that a piece of information can evoke, or the more you can make the viewer feel something with your work, the more readily that information will be remembered by the viewer. Because we are dealing with the emotional storytelling aspect of scientific discovery, it helps to be in the field or the lab where these discoveries are taking place. That way, the storyteller can feel the excitement or grave importance firsthand and even ask questions that the public will likely have, which is where science communication residencies come in. Back to my story, in 2018, I applied to, there we go. In 2018, I applied for and was awarded the Schmidt Ocean Institute's Artists at Sea residency, and it opened up a whole new world of possibilities. I didn't apply thinking I would get the opportunity, having just proposed to make a map of the voyage, which the end result you see here, that's why I've left it on the screen for a while here. I didn't think it was nearly sciencey enough. I thought that they would want something that reflected, more like a infographic-y or something very much more data-driven. But I was chosen, and I was actually scheduled to go on a seafloor mapping expedition, which fit with my mapping proposal. But the time frame was a bit last minute, so I chose a latter date. And I did not know that it was a split cruise featuring both sea trials and the Seeking Space Rocks expedition. I got to spend a week living aboard a research vessel, Research Vessel Falkor, learning about her operations and living life at sea. I was then introduced to the NASA team in mission, hunting for meteorite fragments just off the continental shelf of Washington's state coast, and getting to see the uncut live edition of that deep sea footage that you see on nature specials. And most importantly, I finally got to live out my dream of being on the ocean, which backstory here, I'd wanted to sail on tall ships, but I was a bit shy of it having so many food allergies, but turns out that's not a problem on a larger vessel. So if anybody's wanting to go out to sea, they can accommodate you now. Had I not gone to sea, I would not have been nearly as invested in the work that the NASA team was doing, because I would not have had the depth or understanding, nor felt the thrill of the hunt from being on the team in person. I was briefed before the trip, so I did know what we were looking for and why, but it's exponentially cooler to view it from the ROV control room after having had tea with the engineers and a tour through the ship's engine room, and followed up by snacks about space radiation and planetary protection, which was amazing. The art that I produced from the trip, which is up on screen, was not at all what I had planned to make. My initial idea, let's see, I can pop back, was to, this was one of the sketches that I made, was to make a little more fantastical Tolkien-like map of the seafloor using the data that was picked up from the ROV, the rover, as it was scanning the seafloor. But it turns out that the continental shelf is essentially flat. So it wasn't much of a map, and I had to think on the fly. But instead, I ended up incorporating all of the stories, all of the things that we experienced. So each of the little vignettes around the border is something that actually happened along the trip. And the result is way better than my original plan, and it was meaningful to the crew who all got a copy. I did go on to be awarded more science communication positions aboard other vessels, all postponed due to COVID-19 concerns, but at least there's fun to be had in the future. And I wanted others to have the same awesome opportunities that I did. So I have a list that I put together of all the science communication residencies that include an adventure of some sort. And I do talks like these to tell people that melding your love of art and sciences can be a reality. Usually we are told that we have to choose art or science. Most people choose science because, well, to be frank, there's more money to be had there up front, typically, as a scientist. Art takes a little bit longer to get to being paid well. But I'd like to encourage people to pursue their interests, even though it doesn't always seem feasible, especially financially. It's often a winding road to get to something that you really enjoy, but there's almost always a way to fit things together. And if you just keep trying different solutions, then you can end up being at sea and making travel journals about the ocean. And that is all I have for you today. I kept it short about myself so we could have a little more Q&A in the future.
Can you go back to that last slide? So yeah.
This guy? Yeah, I'll put it up big for you. This was not that guy. That was the cover of my travel journal for that trip.
Nice.
Well, wonderful. Thank you so much for starting us off here today. So, folks, we have two different ways that we can have a conversation. You can either unmute yourself and just speak out, or you can also put anything into the chat box, and I'll be happy to read it out for you as well. And if you didn't want to publicly ask something, you could also send me a private message through the chat box, and I'll be happy to ask on your behalf. I'll start us off if that's all right. Just one of the many things that came to mind here is you mentioned the importance of sort of grabbing people's emotions, and I think there is something. I'm very biased, being a marine scientist myself, but you look at old-timey maps, and there's just something about that sort of that map feel that inspires mystery and exploration. And I'm wondering if you've thought about what it is of being a cartographer yourself. What is it about maps and that particular type of graphic that seems to grab people's attention?
Can you hear me?
We can, yes.
Okay, my screen is like being kind of frozen on Zoom, so I was making sure that it was still working. Yeah, using emotions, or rather using art to evoke emotions, is a really interesting thing. It has to do with a lot of visual design principles and eye tracking and lots of things that people would consider very scientific, but they just don't know that a lot of artists are subconsciously thinking about it, but a lot of us have studied all of these things, and so we know how to work it. The idea of the hand-drawn map, like the fantasy map or the Tolkien-esque kind of map, leaves, how should I put it? It leaves a lot of room for interpretation. So if you give a very, very clear, this is a aerial photograph of this place, you don't have any room for inserting your imagination, for your brain to fill the gaps of what's not there, for you to imagine what is there. So there's actually lessened interactivity with a map that is very precise and very like photorealistic. Whereas when you go toward a drawing, you get to imagine, like, I wonder what this place looks like. Or I'd like to go there to see what that's like. So there's an additional spark of engagement there with a drawn map, especially since a cartographer's job is to, in a sense, be selective. You only show the information that you need. So if I'm creating a map, and it could be By the way, just a side insert, a lot of people are like, cartographers, do those still exist? Haven't we discovered everything already? Any map that's drawn has been drawn by a cartographer. So all of those, I mean, yeah, or even an informal cartographer. So all of those political maps that people have been watching all of these weeks, those are all the product of professional cartographers. So that's what cartographers do. So actually, that's a great example. In a political map, you could show all the data for every single person that voted and exactly where they live, based on their like GPS location, and it would be a mess. Or you could just use the overall voting results and color a whole state a certain color, which is what most people choose to do. So we can be, as a cartographer, you get to be selective as to what you show. And in using drawn and fantasy maps, that's essentially what you're doing as well, being selective and letting the reader fill in the rest of the gaps. Long answer, short question, sorry.
Well, that's great. I'd like to open up to anyone in the participant list. Any questions?
Hello, this is Sherry.
Hi.
And I saw in one of your pictures you were using colored pencils, and I was wondering if you could talk a little bit about your medium and if ocean spray plays a part in what you choose.
Sure, I would love to pop that up again, but my Zoom is like threateningly on the edge of quitting, so I will just leave that. I use whatever works for the for what I'm trying to depict. When I was at sea, I actually hadn't intended to use really color at all, just because I was making notes of things. I wasn't intending to illustrate on the fly, but I had a little more time than I expected. There's a lot of downtime when you're at sea. So I had whatever I had on hand, which was 2 pencils. One you saw the orange, and I had a blue. Those are Prismacolor Coal Erase. I use those actually when I'm sketching illustrations. I use the blue because you can drop it out of a scan really easily to do my like rough under drawings. So I was using the orange I was drawing the what's it called? The line of journey. Oh gosh, it was the path that the boat was following on the map that I'd drawn, just to provide color contrast so I saw what I was looking at. But a lot of the illustrations that you saw were digitally drawn simply because of time. I can color something and make adjustments digitally much faster than I can on paper. And ocean spray, that's an interesting one. It would depend on what I was trying to depict. Like there was an image of a watercolored cat shark, coral cat shark earlier. And I chose to watercolor that just because I hadn't done watercolor in a while, and that was what I grew up doing. So I wanted to do that. But ocean spray, yeah, it would really depend. If I was on a time constraint, I'd do it digitally because it would be easy to change things around and just like, use a certain brush to make splatter. If I was doing it by hand, that'd be interesting. I'd have to use white ink probably, or lots of masking fluid, which would probably end up looking a little, I don't know. Art is an experiment, much like science. So I would probably try about 3 different things before I settled on something that actually looked the way I wanted it to look. Does that answer your question, I hope?
Yes, thank you.
Yeah.
All right, excellent. Other questions from the audience? All right. Well, how about I'll ask one more maybe while people are getting their thoughts together. What's the science artist hybrid community like? Very often in marine biology, I might meet somebody and they have a cousin in Georgia who's a marine biologist, and I'll sort of get, oh, do you know David? It's everyone assumes it's a very tight knit small community, and I sort of have the same impression about the science artistic community where I've known several, and I'm curious how big or small of a community is it, and is there much interaction with other artists out there?
Yeah, that's an interesting one. it's weird. Yeah, we all artists know each other basically. Artist is kind of like science in that you have specializations. And so as a comic artist, I have like 1 degree separation from almost all comic artists, it seems like. Either that person worked with somebody and they're like my mentor, or like it's very, basically we all know each other. And so for science communication and visual science communication, it's strange because I think we all think that there aren't any others out there in a sense. So like once you start engaging with the community, you find out like, oh yeah, you kind of, it's kind of close knit. It's not quite as tight as other specializations, but usually there's like at least like 3 degrees where it's like, or at least I know this person who is also on a ship with this person, that sort of thing. So it's not as close knit since there aren't big visual science communication conventions. There are, but they're not as popular a spot as other things. So we do interact sometimes, but not a ton, not as much as you would think.
Great, thanks so much. So it looks like we have a question in the chat. Sasha, shall I read that? What is one goal you have for your career? Like working for a specific organization, producing a certain body of work, etc.
Yeah, so my career is, I have kind of like dual personalities. So on the fantasy art side, my goal is to move from publishing. So I'm a publisher. I've published several works, myself distributing and all of that, and it makes you tired. I am tired of playing all of the roles now. So my goal for my fantasy work is to get an agent, get a publisher, and transition into someone else publishing my books because I've worn too many hats for too long. On the science communication end, I'm actually more into, I love going out on research vessels and communicating science, but to me, it's more about the communication aspect and inspiring other people. So I'm happy just going out to sea occasionally and then doing talks to inspire people to get out and seek some adventure. My whole thing is about inspiring people to be curious about the world around them. I'm currently running a blog series about navigation and celestial navigation and being more aware of the world around us just as we're walking through the street. And I just want people to just look up and look around. So I'm more on the inspirational half of science communication. It's like my side job to my art. So I don't have big career goals in science communication except more sea time. I want to log more sea hours.
Awesome. All right, well, thanks so much, folks. We're going to have time for questions also at the end. So jot some things down, and you can even get them into the chat box, and we will we'll address them when we get towards the end. Okay. So what I'd like to do is now go and move over towards Steve's portion of the hour. And just by way of a small introduction to that, I want to point out that Steve is part of another talk tomorrow, where he's going to be specifically focusing on bioluminescence and the science behind bioluminescence. So for any of you who want to learn more about that topic, or maybe things that Steve happens to mention today, tomorrow is a great opportunity to catch some more of that. And I'll put a link to the meeting in the chat box here shortly. So I just wanted to point that out. So Steve, why don't you grab the screen and take it away?
Okay. All right, you got my slide up there? Okay. So yes, I'm Steve Haddock. I'm definitely not an artist. That was really awesome to hear about a true artist and to see some of your work, Abrian. But I'm a scientist.
Sorry, sorry. Is anyone else seeing Steve's slide? Because I'm still seeing Abrian's slide. So Steve, why don't you try one more time to grab it?
Go into view options, and then you click on his name.
Let's see. Let's see.
If I can.
Let's see. I'm gonna.
Says it
So I'm
will
gonna,
stop the other person.
yeah. Try to. Do you have the option of stopping the other, Steve?
It said it would when I shared. Let me try.
Yeah. Or maybe, Abrian, are you able to stop sharing your screen? Maybe that will help us.
Yeah, I don't have a. I re-entered, and so I don't have
Oh, okay,
a
gotcha.
button anymore.
Interesting. Okay.
There's two Abriams in my participant.
Yes.
Got it. I'm gonna make both of you a co-host, so hopefully that option should be re-enabled, and maybe that's why we got stuck on your slide. So some people see Steve's side. Okay, there
we
we go. All right, we're back. All right, Steve, why don't you try one more time? Thanks, everybody.
Okay. So yeah, so I'm not an artist, but I'm a scientist, and I think a lot about questions of how to communicate my science. The research that I do, I can sort of divide it into 4 quadrants. One is about these jellies, gelatinous zooplankton, that are really abundant but relatively underexplored. And one thing that a lot of them have in common is their ability to produce bioluminescent light or to have fluorescent pigments in them. So we look a lot at the genetics and behavior associated with those biooptical things. I have a citizen science site.
Sorry, Steve, we're not looking at your screen, so I'm not sure if we're supposed to be or not, but it sounds like maybe you're speaking about things that we're supposed to be seeing on your screen.
You are.
Yeah.
Okay, let's try it again.
3rd time's the charm, I think.
Thank you for interrupting instead of letting me go on and on. Share.
Perfect. All right, we see it now. Thanks, Steve.
I see it? Okay. Sorry. So let me see if my slides advance. Okay. So are you seeing the jelly slide, Kev? Yeah. Okay.
Yeah, looks great.
Okay. So jellies, bioluminescence, and then citizen science through this jellywatch.org page, which has been really a fun way to engage the public. And then also trying to teach people about using their computers more effectively, but as you'll see, a lot of that has to do with graphics. So in general, data visualization people are becoming, I think, increasingly aware of this. This was from the Washington Post. They said that 6 of the 7 most visited stories in their entire history are essentially graphics. And so like Abrian was mentioning those election maps that we were all glued to. I posted a link in the chat to a really nice article that looks at all those maps from a cartographer's point of view and picks some of their favorite different ways to visualize it. Some of the main texts in this are like Tufte is sort of the iconic example, but there's actually a whole bunch of really great books that talk about this, these concepts of how to effectively communicate. And this one on the right, I really like recently. It's using our language, but he actually starts with the basic principles of visualization. And he's written some mapping packages for R and everything. So he's very much got a foot in both the conceptual part and the actual implementation of visualization. This book, this guy runs a pretty popular website. And it's a very colorful book. I don't really recommend this because it violates one of the main principles of visualization, which is that you need to be true to representing the data. So this is one of the figures from that book, and this is the carbon footprint of different kinds of events. So they have this giant wedding cake that has an 11 on it, a ton of shrimp that has a 12, and the salmon that has a 6. If you look at the area of those shapes that he made, this one is 54,000 pixels, this one is 11,000, this one's 25,000. So the size of the images that he put in that page have nothing to do with the actual quantities that he's trying to show. And I think this is one of the things that you have to always be aware of when you're trying to translate data into an image. Another more insidious way that people have fallen prey is by actually falsifying the data. So I'm sure nobody in this audience would do this, but this was a manuscript that I got to review, and I kind of noticed some weird stuff. And then as I looked more and more, I just found that they had essentially cloned, all these cells and just splattered them all over the image to create data that supported their case. And I went back and actually looked at a bunch of these other papers by this set of authors, and all their papers have falsified gels and just completely corrupt research. So nobody do that, please. But I think increasingly we're all dependent on computers in our research. There's really no region. Speaking of Tolkien, there's no region that hasn't been intruded by the need to use a computer. And so we wrote this book, Practical Computing for Biologists. To be honest, it's pretty old now, so it needs to be updated. So I'm not saying this is an infomercial, but just that one aspect, most bioinformatics books talk about programming for, genetic sequence analysis or something, but we have 3 chapters in there just about graphics, because when you publish a paper now, they put so much of the demands are to get your graphics in publishable order. And there's all kinds of concepts about color, different ways to encode color, and different ways to encode images, whether it's as pixels or as vectors. So I think if you're going to learn one thing, if you're trying to get into creating your own visualizations, it's how to work with these things called Bezier curves. There's some fun online games actually that create a little pen tool and let you do exercises to try to get familiar with them. But these are the geometric building blocks of a lot of graphics that we use these days. So one of the things I like to do for my papers is to create icons and little, graphic elements that try to capture. This is from an old paper. My graduate advisor was very traditional, and he was not on board with this first map chart that I made, because he did not like the hammer. This is ways that we were stimulating an organism to test if it was bioluminescent or not. And so we did chemical assays, we did mechanical assays, we even tried electrical stimulation. But he didn't really like the hammer, but I just kept it in there. So that ended up being in the publication. And just trying to create elements that are visual but distinct so that they help you track. The bottom one is from a paper that we're submitting relatively recently, showing different ways of sampling the environment. And so we can reuse these icons throughout the text on different plots. Some of these things are generated. So this is a script that I wrote that monitors some of our underwater vehicles, and it parses out the logs that are coming back and puts them into graphical elements. So at a glance, you can see the status of this little sub that's going around in the bay. And this actually, I counted them up. There's like 23 different pieces of information that are shown on this. And so you don't have to scroll through all the pages and pages of event logs to try to figure out what's going on with the vehicle. I don't know how useful these are to other people, but again, I always like to, if I'm summarizing the functions of bioluminescence, for example, I like to make a little scenario that kind of illustrates that, as well as the text to go along with it. So these are some of the functions of bioluminescence. More recently, we did an op-ed and a paper about the effects of mining on the deep sea water column. So I just created this little scenario of like before and after of how the organisms are interacting under normal times and how they're interacting when deep sea mining plumes are potentially interjected into the water column. And as I said, I'm not an artist, but on this one, I really kind of was emotionally connected to this project. So I made a little sketch to accompany it of just like kind of conveying the emotional aspect of this research. We're trying to. You have to make a lot of different choices when you're trying to visualize a large amount of data. So this is a matrix from a paper that we did with Anela Choi, where there's predators across the top and prey down the side. And so each column is what that predator eats. And one way to show it is here, and you can get some information out of this, but you can also show it as like a network diagram where the connections are coded by the predator color. And this shows you, one, the complexity of the situation, but you can also zoom in and focus on, here's one kind of a comb jelly, it's kind of jellyfish that eats a certain kind of polychaete worm. So there's very specialized relationships that are happening in this network. And then there's also very general things like one of Kevin's favorite organisms here. We found that it ate 22 different kinds of prey in our study. So you partly have to work with graphics and ability to generate graphics with computer, but photography, and Kevin can attest to this, pretty much everybody who studies jellies ends up being a photographer as well, because they're too fragile to preserve them properly. So we need to take a picture, and that ends up becoming our preserved specimen, essentially, to figure out the morphology. There's a lot of considerations with photography that people struggle with. And if you look in a lot of papers, you'll see microscope images where it's like a yellowish, it looks like a sepia tone image. And that's all a challenge with setting the white balance of the camera to match the white balance, the color signature, color temperature of the illumination you're using. So these were pictures that I just grabbed off one restaurant website. And you can tell that they, they set up these pictures, but then they took them, like in one case with the strobe. This is taken with a strobe with the color balance settings wrong. This one is, whoops, sorry. This one is taken properly. It's like in daylight with daylight settings, but the rest of them are taken like with incandescent lights and the wrong color settings on their camera. So some of them look more appetizing than others, I would say. Another aspect of photography that you have to kind of deal with, I think, is depth of field. You can use this like on your iPhone when they do that portrait mode. It essentially is putting the focal plane on the person's face and fuzzing out the background, creating this artificial depth of field to highlight the subject. But as you close down your aperture, your opening on your camera, the depth of field can increase. So sometimes you can use this to your advantage, and sometimes you can use it for artistic effect. So illumination is obviously really important, but we also can use photography to capture things like fluorescence. So this is a case where we're using a blue light to illuminate this same species, put a yellow filter on it. So you're only seeing fluorescence, that it's excited by that blue light. And it gives you a different perspective on a lot of what's going on. There's actually, I forgot to put a link to it, but there's a video we made about this jelly called Lindius, and it has a lot of fluorescence video and images. I can post it after. And then we can also capture bioluminescence. So here's a ctenophore in regular white light image. But if we do a time exposure, you can see the light that the organism itself is producing coming out as bioluminescent light. So as Abrian mentioned, also people think that a photo is like the gold standard of documenting something, but really when you create an illustration or a map, you can choose what kind of information to highlight, and you can reveal things that you can't see even in a photo. And so to go from a photo to an illustration, sometimes we'll just take one of the photos as a reference and then essentially trace it and kind of stylize it to form the illustration that lets you highlight the features that you want. Sometimes I've gone a little bit further. These are stinging cells from this kind of organism called a siphonophore, stinging cell batteries. In these cases, I made a 3D model of the internal windings of it because I'm really not a good enough artist to like draw that just from scratch. And then I can trace that 3D model and create the final layout that appears in the manuscript. So we've used this trick a bunch of times where, I just am not good enough to get the angles of these arrows to look realistic if I don't have some kind of reference. So I'll create a 3D model and then trace it and make it into the illustrated version of it. This is another example from a paper that is just coming out. These are the sticky cells from one of the ctenophores, those cone jellies. So my co-author drew up this thing of the features that we wanted to show. But then I wanted to like slice through it to show the inside and the outside at the same time. So I made a 3D model of it, which we could then trace and, you know, create the final illustration from having a realistic and accurate perspective. And then one last aspect that I've used is animation. So this is, I was doing an analogy of amino acids as Legos, so that you have these building blocks of proteins that can go together to form three-dimensional structure. But the linear sequence of those things can have variations at different sites that are reflected in the final organism, the properties of the final organism as it's put together. So that was kind of working by analogy using animation. And then also we have a video about these by-the-wind sailors where I think of them as sort of sailing on the underside of the ocean. So I did an analogy with that Pirates of the Caribbean movie where they sail underneath the surface to show kind of how those jellies live at that air-water interface. So that's just some general examples of ways that I've tried to use graphical concepts to communicate science.
Fantastic. Folks, as we did last time, you feel free to unmute yourself to ask questions of Steve, or you can also put any questions in the chat box as well. I'll start if that's okay, Steve, just while others are getting their thoughts perhaps typed out. I'm curious about I think of you as being kind of a leader in this this push to make the scientific illustrations at one more friendly to the reader, but also sort of more informative by not shying away from being a little bit more icon or cartoony driven. And I'm curious, over the couple decades now of your publications using this, have you encountered a shift in the kind of editorial opinion on these things? Are they more welcomed now than they used to be?
I don't really know. I mean, I would be curious to hear more back from people. I think they do appreciate it. And with my co-authors, a lot of times, like we'll have a sketch of a figure like I showed, and then I'll try to iterate that a couple times back and forth with them. And it seems like, you know, everybody agrees at the end that may be a better way to do it. But for example, on the functions or on the deep sea mining little thing, I don't know actually how many people like me appreciate the little cartoon of it versus an actual sentence or paragraph explaining it. There's definitely been a shift. I mean, obviously with photography and the advantages and not of photoshopping, but publishers have really pushed the desktop publishing labor onto the authors these days. So you kind of have to do it. And I think using R and ggplot in particular, like has pushed a lot of people into good habits because the defaults on those are usually a lot, I would say, better than what they were before. It's harder to get lured into making a 3D pie chart of your data.
Great. Well, we have a couple of questions that came into the chat, but I'm going to ask Anna first to say it out loud.
Oh, hi. I don't even remember what I wrote. I think I was thinking about how talented you both are in trying to think outside the box. What is it that you see yourself doing in a few years that you've never been able to so far? Maybe use what you have in your fantastic brains and do something totally different, or maybe the same thing, but in a different place. Any thoughts?
I mean, from my perspective, I would like to get a bunch of this species descriptions that we have backlogged done, and that requires, I'm just hiring this amazing illustrator named Nick Bezio, actually, to do a lot of the drawings, so that's been a bottleneck for me, is he can do it a lot better and a lot quicker. So I would like to get a bunch of the publications out on some of these deep sea species and have, you know, really iconic illustrations that that Nick can create to accompany them. I don't know if that's big picture enough for you, but that's been hanging over my head for a while.
Abrian, any comment to that question?
Yeah, for me, it looks like in the chat it was a project that I'd love to work on, but haven't been able to yet. Way back thousands of years ago when I was in college, I used to do gilding, so like gold leaf, and like I would do a lot of manuscript work. And so it was always my dream to do an illustrated bestiary, like very old school, but maybe, and I had no preference fantasy over reality, but it would be very cool to be able to do an old school illustrated bestiary of things that exist,
That would be
but
amazing.
you know, might be like deep sea creatures. I think it would be amazing. I also want to be part of the 2030 initiative to map the world's oceans and do a really cool update of the one of the first ocean maps. I don't remember the lady who made it. Sorry, I'm blanking on her name, but I've always wanted to do a really, really cool detailed map once we get all the world's oceans mapped, which they're pushing for 2030. We'll see. That's the goal.
Fantastic. We had another question come in from Jessica. Jessica, would you like to ask it?
Yeah, I just thought that trying to take pictures of jellyfish would be extremely challenging for multiple reasons. And so I was wondering if each time there's a lot of trial and error, or if, you know, over time there are just techniques that you are like your go-to, like I know it's going to be one of these two settings and I just need to figure out which one it is.
It's sort of the latter at this point. I did write a little article for Make Magazine about photographing jellyfish a while ago, but it's getting the lighting set up right with the strobe. I like to use one strobe because I think it makes the contrast and the highlights better. If you have two strobes, it kind of flattens everything. But then actually on my camera, I have like two user presets, and one of them is for bioluminescence or fluorescence, and the other one is for the tank shots. And I think it really is the latter. You have the aperture set and the exposure set kind of in the ballpark, and then you just adjust depending on the particular scale of the organism. But it does take it takes trial and error, and I mean, Kevin can speak to this too, but once you get it, like I've had people come on cruises with me and they're taking kind of the normal shots when they get on, and you just give them like a few pointers, and by the end of the cruise, their images are, you know, looking really nice. So I think it's not that complicated, but there's a few pointers that can help.
Great. We had another question that came in from Stephanie. Stephanie, would you, I don't know if you'd like to unmute yourself and ask it?
Yeah, I was just curious about what you each like hated the most about your job, and then what was like the best part of your job, like that makes you excited to get out of bed in the morning. So like from the most mundane to like the most exciting, what are those aspects for each of you?
Abrian, do you want to go first on that?
Sure. So I'm a weird species. I like checking things off my list. I like getting things done. I am trying so hard to learn to enjoy the process, and that's probably why I talk so much about like going on journeys and looking around at the world around you, because I am not good at enjoying the process of things. So I like when I finish things, you know, I like seeing the final result, like, wow, that was fun to do in hindsight. And then moving on to the next thing, because I'm probably already working on it. What is the worst part? There, I mean, like, as you said in your comment, like there's always the, you know, making phone calls and applying for grants, although you get used to that. Like if you do, a challenge to all of you. I spent a year applying for something, or at least taking action, like following up on an application for something every week for a year. So I ended up sending 35 original applications. And heard back from half of them positively. So like with a 50% strike rate, like why wouldn't you apply for anything? But that was good practice and just doing it over and over again. So that is not actually a big deal. I think the worst part is probably when you hit a snag and you're not sure what to do, and then you have to think through whatever the new solution is going to be. That can be extremely frustrating because it's like, well, I already tried my best and it's not working. So I don't know what my new best is. Like, I don't know what the solution to this is. But that's true of every job.
What do you do when you hit that, Abram? Do you have people you can bounce stuff off of it?
I do. Sometimes they're in a similar ballpark to me because I'm terrible at asking for help. So I've usually like exhausted all of my brain power and resources. And usually I'm like, they're like suggesting things that I've already thought about, but it does help to just say it out loud. Sometimes you get the solution just by asking the question out loud.
That's what I was thinking. Yeah, with programming.
Yeah. Yep. So I do definitely talk to people, and I will, you know, try, you know, reading a new resource or just doing something else entirely and hoping that it inspires something, which there are very few eureka moments, but they're not impossible. How about you?
I mean, for me, the frustrating part is trying to get funding, because all my funding that I'm trying to get externally from NSF or whatever is to support students. And I feel like that shouldn't be such a struggle. I won't even tell you how many rejected proposals I have before we got 2 in a row funded, which is amazing. But I mean, I had like 14 before that were rejected. So it just gets discouraging. And the timing of it, where you're applying for it, but you're not going to get the funding till here. And then the grad students have to apply and they can't start till here. So that to me is the most frustrating part, I guess. But I mean, the rewards are obviously like seeing these organisms bioluminesce for the first time, or being able to capture it on camera. Now we have video cameras that can film in color what you can see with your eye. And so being able to capture some of the sequences that I previously had been only able to see like from out the porthole of a submarine or something, that's really, really pretty fun. And also working with people like the engineers and my collaborators on it, and just having like smart, talented people everywhere you turn is really fun.
Great. We had a question come in asking about the challenges of working with just pictures of jellyfish rather than live species. Maybe you can, Steve, you can talk a bit about how much time do you spend with the live animal versus the representation of that live animal that perhaps you've done via photography, or these days even 3D modeling?
Mm-hmm. I mean, a lot of the observations that you make are initially done visually, like under the microscope or by eye looking at the animal, depending on how big it is. And then, you know, you capture notes of like how this part of it connects to that other part, or which way this little tube routes. But the thing is, with the deep sea animals, they're living at like 1.5 degrees Celsius, or, you know, maybe 2 degrees for some of them. So the more time you keep them under the microscope, you can just see them melting before your eyes, really. And so I lately have come to just try to take as many close-up overall, you know, overview photos of the features that I know I'm going to want to look at later. And it is better to capture them that way, because when you're on a cruise, you're usually sleep deprived and you can't necessarily, you know, make all the accurate observations that you want to.
Great. One more question here I'm going to ask both of you to sort of reflect on the use of computers in your versions of science communication. Steve, you mentioned the very fascinating idea of creating what I think for somebody who's not into computers, a 3D model of something seems like very complicated. And then you go ahead and turn that back into a drawing, which seems, I think, in a lot of people's minds to be sort of a reverse of what maybe people would expect. And likewise, Abrian, you talked about sort of the advantages of using digital artwork workflows in terms of speed. Maybe you could both reflect on sort of the nature of having those sorts of tools at your disposal, what they allow you to do, or maybe things you wish you could still do but are having the technology is not there quite yet.
I mean, for me,
Steve, yeah.
it's really become so inseparable. We try to generate as much of the image programmatically as possible. So for that food web diagram, for example, that was created in R. We posted the code that generates that as part of the supplemental materials for the paper. But then we always take it into Illustrator or whatever and like fine tune elements or highlight certain things after the fact. But even with photography, it's like, when I started out, it was like in the dark room trying to like dodge and burn with the enlarger on a black and white thing and using rub-on letters and all that stuff. So it's the ability to modify or enhance your images or clean them up or annotate them afterwards it's completely game changing. We can also shoot literally thousands of shots, where before you had film on the Cruz, you didn't know if the exposure was right or not until you came back and 2 weeks later got it developed and you were like, oh man, these are all blown out, or these are all too dim. So that aspect of it has completely changed.
Yeah, for me, it's definitely a speed thing. Just in, as Steve said, being able to change things. It used to be, if I'm drawing a person and the head's just a little bit too small or big, then you just have to redraw the whole thing. Whereas if I'm working digitally, I can just select it and move the little handles. Perfect. I'll fix. And so I will, if nothing else, I will work roughly setting things up digitally oftentimes and then switch to traditional. I work faster traditionally, but again, I can't readily change things as easily. But even if I work completely traditionally, all on paper, whatever medium, I still, if I want to be able to share this with other people, and I'm not just going to go make copies on the copy machine and hand them out, I still have to digitally transfer it into a digital medium and then share it that way. So either way, I'm not getting out of using the computer. As far as technologies that aren't developed yet, I'm sure I can think of something that's very, very specific. Like I wish that Photoshop was able to move this thing without moving that thing at the same time. Or, that 3D modeling was a little bit less wonky and a learning curve, even when you're using like SketchUp. But for what I need to do, most of the technology is already in place. Scanning could be better. I don't know why it's not. But aside from that, I have all the tools. It's just whether I decide to use them and how effectively.
Oh, awesome. Well, I think we've all seen a lot of great examples this morning too of how both of your use of these tools is really also matched by the inspiration and the artistry that connects into them, right? And so I want to thank you both so much for joining us here and allowing us to record this for all the folks that weren't able to make it. Just one last plug for everyone to check out the STEAM 2.0 website. I'll put it over here in the chat box as well. And encourage everyone to go in and take a look at all the interesting bits of art that people have contributed from all around the campus in lots of different ways, from poetry to drawings to a jellyfish made of trash. This was just for Steve's honor. Please take a look at the STEAM website, and please also note that we have still an upcoming lecture that is going to be happening on the 24th at 11 a.m. So thanks to everybody, and thanks again to Abrian and Steve for taking time out of their very busy days to spend some time with us this morning. And thank you all for joining us. Take care, everybody.
Thanks, Kev.
Thank you.
All right.